everyone. My name is Jerry Wilson, and I'm the Executive Director of the Pediatric Cancer Research Foundation. And I would like to welcome all of you to one of our first panel discussions. We're going to be doing a series of these. This particular discussion is going to be talking about patient care in the pandemic. I would like to go ahead and just let everyone know that this call is being recorded, and we will have it by the end of the day that we'll be able to send out to you, and you can forward it to anyone you like, because there's going to be some great information shared by some of our top and leading researchers and one of our social workers at the hospitals that we support. I'd like everyone to know that there are two features at the bottom of the screen. There is a chat feature, and there's also a Q&A feature. If you would like to talk amongst yourself, please use the chat. And if you'd like to do any questions for any of our distinguished guests, go ahead and put them in the Q&A. I will be saving them until the end of our talks, because we just have a limited amount of time today, and we've got a lot of great material we want to get through. All of our panelists will be able to see your questions also. And with that, I'd like to go ahead and just get started. A lot of you on the call know PCRF because you might either be a board member or a family that we've supported, or maybe this is your first time getting to know us, but we have been around for 39 years, and all of the panelists on the call are all funded by PCRF and have been for quite a few years. We have a new person also, but we're super excited about that. And really, we've been in this sector really just trying to fund top and leading key researchers, all to eliminate childhood cancer. And for those of you who aren't aware, we are one of the most underfunded diseases by the federal government. And without the support of organizations like ours and a lot of our collaborative partners who also support many of these researchers, we wouldn't be able to do the great research that we do. And so I want to share with these researchers, because this year, like next year, I'm sure we're gonna take a hit, and all of these researchers are going to need funding. We want to do our best to get our message out there and awareness for all of them. So with that, what I would like to do is go ahead and introduce our first speaker, Dr. Brian Crompton. There he is. Dr. Crompton is at Dana-Farber, and he specializes in Ewing sarcoma. I had the pleasure of meeting Dr. Crompton back in, I believe, 2012 or 13, when we were doing an event with the Boston Red Sox. And John Lester, one of their big lefties, who's now with the Chicago Cubs, is a pediatric cancer survivor. And Dr. Crompton came to the event, and I just got a chance to meet him and get to know him over the years. Some of Brian's favorite things to do are scuba diving. He loves to scuba dive with sharks. I'm not sure that many people would say that, but that's something up high on his level. And then, yeah. And then I also wanted everyone to know that I asked each one of the researchers just a couple of questions, and that he knew that he wanted to get into research when he was in junior high school. So this has been a lifelong passion of his. So that is Dr. Crompton at Dana-Farber. And then I have Dr. Elliot Stieglitz, who this is our second year funding his research. He is at University of California, San Francisco. He is working on juvenile leukemia, which is new for a funding platform for PCRF. One of his favorite things to do when he's not working is to travel, and I know that our environment right now is not kind to that for you. He also let me know that he's one of the top Scrabble players in his family, so next time I have a report to read, I might let you work on some of those words for me. He has been at UCSF since 2011, and this is our second year in funding the work of Dr. Stieglitz.
Pleasure to be here.
Welcome. Then we have Dr. Ashley Plant. I first met Ashley when she was at Chalk Children's Hospital here in Orange County. She was only here for a couple years before she got whisked away to Chicago to Lurie Children's Hospital. She is a specialist in DIPG. They are incredibly lucky to have her. She is also our Will Irwin Memorial Fund recipient. We took care of a young patient out here, and the family has been incredibly gracious and is a big supporter of Ashley's. She realized that she wanted to be a researcher when she was working on brain tumors. She learned that the worst brain tumor prognosis had not changed in over 30 years, and she wanted to throw some of her efforts and energy behind that. When she's not working, which I'm not sure when that is, like the rest of them, she loves bungee jumping and skydiving. So I think we have another adrenaline junkie here. Welcome, Ashley.
Having me.
And then last but not least, we have Katie. Katie works at Lurie Children's Hospital as a social worker with children with brain tumors. She's been with Lurie for over 5 years. She knew that she wanted to work in this field when she was early in life. She envisioned the world would become, she envisioned she would become an English teacher when she first went to school as an undergrad. She had an opportunity through a AmeriCorps program to work with seniors in an assisted living facility and health and wellness program. Many of them were immigrants to the US and shared the most amazing stories of their youth and the transitions in their life. Hearing their stories and bearing witness to the resilience and commitment to a lifelong healthy began to change and how she could interface and change the world in working with kids. So we welcome you, Katie, very, very much, and thanks for what you do behind the scenes.
Thank you.
And with that, I am going to turn it over to Dr. Crompton and let him start his slides.
Swap display. So I'm looking at. Can everybody see the projected one, or are you seeing the broken up one? Does it look okay? Slides look okay? Yeah.
Okay.
Well, thanks so much for the opportunity to be here. I want to talk a little bit about the research, but you know, I think the length of time is a little short to get really deep into science, which of course I'd be more than happy to talk to any of the audience members about in more depth at any time. But I wanted to really highlight specifically how PCRF as a foundation has really accelerated or amplified the research we're trying to do. So I'm going to sort of almost give it, I actually will give a little bit of a timeline of support over the years as a way to really exemplify how important foundation funding is to the development of a research program. I have one slide here about Ewing sarcoma because it's the primary interest of my lab. And just so people understand the background and the importance of working on Ewing sarcoma, it's the second most common pediatric bone cancer. It affects adolescents and young adults. And the treatment, of course, is with, as is true for most of our pediatric cancers, is with intensive chemotherapy combined with either surgery or radiation. And this is, you know, the treatment comes along with some significant risks of long-term toxicities. The survivals, you know, has definitely improved significantly over the decades and now is probably around 70–75% for patients with localized disease, but at least 20% of cases present with metastatic disease, and that's much harder to treat. We also know after disease comes back after treatment, if it does come back, that again, it gets much harder to treat. And we honestly have no idea why. We don't know why recurrent disease is so much harder to treat than newly diagnosed disease. And so we're interested in new treatments and understanding how existing treatments, how patients' tumors respond to existing treatments and how they develop into resistance. And so I'm just going to kind of give you an overview of how we're sort of approaching this in my group, but really wanted to integrate in here this timeline of Pediatric Cancer Research Foundation support, because I think it's so important to understand that, you know, progress into developing a research program isn't possible without, you know, serious investment and relationship building. And so back in 2013, before actually, while I was still a postdoc, we had published a paper about a new target in Ewing sarcoma. And soon after that, we got our first grant from PCRF. That led to the next level work, the follow-up studies, which eventually matured into an NIH grant. And that NIH grant was my career development grant that really catapulted me into eventually having my own independent lab. Without funding from PCRF and others at this stage, I would have never gotten to this next stage. That led to another paper and more funding and a whole nother project. And I'll touch briefly on each of these steps of the research as we go through the slides. But because this NIH funding came through for this initial project funded by PCRF, PCRF graciously took on a second project that is almost kind of similar. It was pretty actually unrelated to the previous project, where we started to adapt genomics to really clinical management. And that has merged into, well, that resulted in an early publication that resulted in PCRF funding additional research, really evaluation of a prospective study to really validate liquid biopsy. And that has catapulted us now into doing liquid biopsy work across multiple pediatric diseases. And while PCRF still fundamentally funds the Ewing work, we now have additional NIH funding for osteosarcoma. We have Department of Defense funding for neuroblastoma. And we have Hyundai Hope on Wheels funding for rhabdomyosarcoma. So I really wanted to put this together because I wanted to highlight how, like, you know, it really is like planting seeds. Like, it grows, like if you plant seeds early in young investigators' lives, it can grow into things that are beyond what any one foundation can really support. And that's definitely been the case for me. I'm going to talk briefly about each of these just so people have a flavor of what we work on. This was a study that we did really while I was a trainee, and we found that focal adhesion kinase was this highly expressed enzyme in Ewing sarcoma, and that when you inhibited it, it really drastically reduced the rate of tumor growth in mouse models of Ewing sarcoma. And that merged into this next project of saying, well, we know that single agent therapy using one drug to treat a cancer never really works. So we need to find interesting combinations to go along with this focal adhesion kinase inhibitor. And this work was partially funded by PCRF in the early days of this follow-on study. And then this was the part of the project that was funded by that first NIH grant my lab received, or I received, where we actually worked with a group called NCATS at the NIH, where they do drug screens. And this is a picture of a robotic arm where they're adding, multiple drugs to plates with cancer cells in them. And we look for things when they combine, they have more than just the additive effect. So if drug 1 has X effect and drug 2 has Y effect, we get more than just X plus Y, and it's called synergy. So we were looking for these combinations of drugs that resulted in synergistic killing of Ewing sarcoma cells. And we found these aurora kinase inhibitors combined with FAK inhibitor really had that synergistic activity. These dots up here imply synergy. And when we gave mice the two drugs combined, they did better. They were less likely to die of their Ewing sarcoma tumors when given the combination than if we're given either drug alone. And that actually prompted us to try to write a trial, a phase 2 study to look at aurora kinase inhibitors in Ewing sarcoma. And we got so far as to make a whole protocol. It was actually fully written. We had involved a drug company that had an aurora kinase B inhibitor. And then something happened that is not that uncommon in pediatric cancer, is that the drug company decided this wasn't a drug that they were going to pursue for an adult indication, and so it's not going to become a drug. And because they needed an adult indication to justify the financial investment in converting it to a drug, and since they didn't have that, they killed the drug, and now we don't have this study. And that kind of pissed me off and got me frustrated. And I said, well, okay, then if this is going to periodically happen, we can't have one or two combinations. We have to have a lot of combinations, because maybe if we have a lot of combinations of things that are emerging as new targeted therapies, we'll have our best shot that some of those drugs will actually make it into clinical development and actually become commercially available drugs. Then we can use them in rare pediatric cancers. And so we did another new screen, actually almost a little less than a year ago, really during the winter of 2019 into 2020, where we took 8 cell cycle inhibitors, and I think it was 15 or so kinase inhibitors, and we did every possible combination. So we did 136 drug combinations at multiple doses in Ewing sarcoma cell lines. And we have, at least a dozen combinations that look interesting there. And so we're now in the process of trying to validate that, and that's where this project currently stands. But I think it's exciting that we may be able to take 2 classes and not care so much about which drug we're picking now, but actually figure out what 2 classes of drugs really combine synergistically, so that if any of them become drugs, we have a real shot at getting it to our patients. And then I mentioned that around 2016, Pediatric Cancer Research Foundation started to fund another project that really was one of the starting projects for my lab specifically, where we started to look at what's called circulating tumor DNA or liquid biopsies. And the idea here is that it turns out that tumor cells, tumors actually shed little bits of their DNA into the blood. And so now that we have these extraordinarily sensitive sequencing technologies, we can actually detect the presence of tumor DNA from a blood sample. And we can basically now almost profile the tumor sample itself, like identify the mutations that are driving the cancer by just a blood sample, which, I mean, you know, if you asked me about this 10 years ago, I'd say it was like science fiction, but it's real now. And my lab does a ton of this. Our first project was focused on Ewing sarcoma. And we developed a brand new liquid biopsy assay specifically to find the mutation that drives Ewing sarcoma. We succeeded in doing that, and we even were able to look at a cohort of patients with Ewing sarcoma and show that if they had a lot of this circulating tumor DNA, those patients did much worse. And so now we've taken that finding and we wrote a multi-institutional study where we're validating that in a new set of patients to say, does this still hold up using modern therapy for Ewing sarcoma, and to prove that this is now a technology that we could use to say, even though these two patients look like they have very similar disease and should have the same risk, this patient actually is much more likely to do well, and this patient might not be cured by therapy, and we should change therapy. So we're validating this as a biomarker. We also have questions about understanding tumor evolution and how relapsed disease results in treatment resistance. So that's just sort of a really quick overview of what we're working on in the lab. The last thing I wanted to just touch on was what Jerry alluded to about, you know, the impact on COVID, both on, you know, funding and research operations. We were unbelievably lucky in my lab that we basically got this data from NCATS, and we got a whole bunch of sequencing data from our prospective study in liquid biopsy literally before my lab shut down for 3 months. So we were lucky in the fact that we had sort of done a whole bunch of the experiments we had proposed to do, and were able to analyze all that data. But, you know, for the postdocs in my lab, it was super stressful because they couldn't touch pipettes. They were at home working on their computer when they really wanted to be doing experiments. And that's going to honestly set back their maturation into independent investigators working in this field. And then I think the other thing that is definitely true is that right now we're already seeing how COVID is impacting funding from donors who give directly to our labs and foundations. There's just going to be less money for grants out there for sure for the next year, maybe longer. And it's only a matter of time, if the economy continues to struggle, that the NIH will also have less money to fund work. And so I just can't stress enough, like you can see what seed funding does to like sort of build up a whole program. I can't stress enough how critical it is right now that, you know, if you believe in these causes and you trust the foundations you're affiliated with to distribute the money to the best projects, that if you have the means, and not everybody does right now, but if you have the means, we really, the community is going to need people like you guys to double, triple down. And so, and I also just wanted to sort of obviously express my sincere gratitude to PCRF, all of you out there who work so hard to raise the money that we need to make those research experiments happen. And there's a whole bunch of people in my lab and other labs that we collaborate with that make this stuff possible. But I'm sure I've probably exceeded my minutes, so I'm gonna hand it back over to Jerry.
Thank you, Dr. Crompton. No, it's been our joy to work with you, and I know it's like, you know, speed research, and I appreciate you taking the time to do this. And again, I know all of these researchers and panelists and social workers would be happy to follow up with any of these questions. You can reach out to me, and I can connect you guys up. But with that, Dr. Stieglitz, I will turn it over to you.
Perfect. Thank you, Jerry. Really excited to be here. Brian, that was fascinating work. Glad I got to listen. I think you'll hear some similarities in what I'm about to present, and also some slight differences in terms of what we're studying and how we're studying it. So as Jerry mentioned, I'm in my second year of funding from the Pediatric Cancer Research Foundation, and I study a type of leukemia in children called Juvenile Myelomonocytic leukemia. That's a mouthful, so we'll just use the abbreviation JMML. And so when we think about leukemia, I'm just going to take one slide to introduce the disease. inside of every single one of our cells, and we have billions of cells in our body, there are letters of DNA. And these letters of DNA are actually the instructions for what every cell needs to do. And every time a cell grows, it needs to copy its DNA, make an exact copy, and give it off to its daughter cell. And so here we were looking at the different types of letters of DNA in the original cell. And now the cell is about to copy itself and make an exact version for its daughter. But what we can see is that the fifth letter over here, instead of a T, it's actually a C. And this is what we would call a mutation. Now, inside of every single cell, there are billions of letters. So you could imagine that when a cell is copying itself, sometimes there are errors. And if that error occurs in the wrong spot and causes the cell to grow faster than the original one, it will eventually turn into cancer. So within the blood, there are three different types of blood cells. There are red blood cells that give people energy. There are platelets that help people from stopping from bleeding. And then there are white blood cells that fight the infection. Almost all of the cancers of the white blood cells almost all the leukemias are cancers of the white blood cell. And in general, in the United States, there are about 4,000 cases of leukemia, which again is a cancer of the white blood cell in children. So this type of leukemia that I've been studying, which is called JMML, affects very young children. The average age at presentation is only 18 months. In contrast to other more common types of leukemia, this seems to be very aggressive on day one. When these patients come to us, the leukemia cells have left the blood and have entered other solid organs like the spleen, the liver. It can enter the lungs, which causes breathing complications, and sometimes even the skin, causing a rash. And in general, compared to the more common leukemias, the outcomes in this disease are very poor. We need to use the most intensive treatment we have available to us, which is something called a stem cell transplant, where you kill off all the normal and abnormal blood cells and then replace it with somebody else's immune system. And here we see a picture that I think recapitulates a lot of these aspects of the disease. We see a rash that no one would suspect is actually leukemia, but when this was biopsied, we actually saw leukemia cells in this very young baby. And this patient received multiple therapies and initially did not respond, but eventually was able to get to a stem cell transplant. So when we think about why are these very, very young babies developing leukemia, we know that the initial cause, as I mentioned, is a mutation or a change in the DNA. In the initial figure I showed you, there were only 10 or 12 letters. What gets more complicated is when we want to look at all the letters of DNA inside of these blood cells. Remember, in each blood cell, there are billions of letters. So how are we going to find what actually causes this leukemia? So the way we do this is we're trying to find this needle in the haystack. And just looking in the leukemia cell would almost be impossible. So what we do is we actually take two different cells from each patient. So from that same patient on the previous slide, we also take a normal cell from inside the baby's cheek, a simple swab, and then we compare it to the leukemia cell. And basically what we're doing is looking for something that appears in the leukemia cell that does not appear in their normal cell. And so in this case, what we can see is that the fourth letter is actually an A instead of a G. The only way to know this is by comparing the leukemia cell to the normal cell, because every single person on this call, if we were to look in their blood cells, there would be tens of thousands of changes amongst us. That's what makes us different. But the only way to know that a change is actually causing leukemia is to make sure that it's not present in the normal cells, but is present in leukemia, and that's what caused it to develop. So as Brian mentioned, when I was a postdoc in my mentor's lab, we carried out an experiment in 2015 that looked at 100 different patients with this disease. And over here on each column is a different patient. And on each row are the different genes. And what was really surprising about this experiment, testing 100 different patients' leukemia cells and normal cells, is that almost every single patient had a mutation in one family of genes that we call RAS, R-A-S. And so anytime a patient has a mutation, there's a box in that gene. And so what we can see is out of the 100 patients, 95 of them had a mutation in one specific family. Now, this was really important and it helps us make a diagnosis. In the past, it was incredibly challenging to make a diagnosis of this disease. But now with this next generation sequencing, we're able to make a diagnosis in at least 95% of patients. But the question that we still have is half of the patients with this disease are going to do incredibly well and will be cured of their disease with a stem cell transplant. But the other patients are going to do very poorly even with a stem cell transplant. And so we don't want to give therapy to patients that are not going to benefit. But we need to know who is going to respond and who is not before we start that treatment. So what we've done is we've started to look at something called DNA methylation. And DNA methylation is different than the mutations we talked about before. So on the first slide, I showed you that this patient had a T that turned into a C. And we now know that can cause leukemia. Now, the process of DNA methylation is normal in our bodies. Across millions of cells, across millions of letters of DNA, there is a methyl group, a carbon and 4 hydrogens that sits on top of letters. Now, if you were to take a typical approach and just read the letters, this actually looks very similar to the original sequence, but there's actually a methyl group on top. And the presence of this methyl group really does change how the cell operates. It's the equivalent of a mutation, but you cannot just read the letter because the letter T looks the same. You need to use a different approach that looks for the presence of this methyl group on top of the letter. And the process that this refers to is something called epigenetics, epi meaning on top, and genetics being the letters of DNA. So DNA methylation is an example of epigenetic modification where the letter doesn't change, but something on top of the letter changes. So here's a complicated figure that shows, again, patients are on the columns on the bottom. And here we're looking at DNA methylation across 80 patients with JMML. And in general, blue means that there are very few methyl groups. And if you see the red, like over here, that means that there are many methyl groups. And to our surprise, the patients with very few methyl groups did phenomenally well. Those patients survived more than 90% of the time. And patients with a lot of methyl groups only survived 20% of the time after transplant. In addition, what was even more surprising is that there's this rare phenomenon called spontaneous remission, where patients' leukemia just goes away on its own without any treatment. And when we looked at all the patients that had spontaneous remission, they lived very close to each other. And their DNA methylation signature was very similar to each other. And so we think for the first time, we have a way to predict which patients are going to spontaneously resolve, meaning that they don't need any treatment whatsoever. So the goals of the PCRF grant was to establish a DNA methylation test in a clinical lab. And like Brian mentioned, to get funding for a clinical trial, which can cost over $1 million, is incredibly challenging. And so sometimes what you really need is preliminary data to support a trial to go forward. And so we accomplished the first part of this PCRF goal, which was to change this DNA methylation assay into something that anyone across the country can order. And now with this in place, we're actually ready to run a clinical trial. Initially, we had proposed doing a registry, which means that we'll just collect samples and see how patients do. But we've made so much progress that we've actually developed a full clinical trial that's going to test the ability of DNA methylation to predict outcomes in patients. And so here we see this is an international collaboration between the United States, Germany, and Japan. There are almost 300 patients in this cohort. And in this one figure, we see that the patients with low methylation, none of these patients relapse. They have incredibly favorable outcomes. And in contrast, the patients that are high methylation or have lots of methyl groups, only about 20% of these patients are alive after 5 years. So the goal of our study is to run a clinical trial where we're going to do DNA methylation analysis in a clinical lab. And the patients with very few methyl groups, they're going to do very well with or without therapy. So they are not going to receive a stem cell transplant for the first time. And in contrast, the patients that have a high amount of methyl groups, we know these patients don't do well with just a transplant. So we're going to give them targeted therapy plus chemo and then take them to stem cell transplant once they're in remission. And we hope that this will improve outcomes for both this cohort and for the first time skip a stem cell transplant in patients that don't need it. And so in conclusion, JMML is a rare disease, but it is very aggressive and requires usually intensive treatment. Almost every patient has a RAS mutation. We are now able to diagnose this in a clinical lab. And DNA methylation can tell us on day one who is going to do well and who is going to do poorly. And now we are going to start tailoring the amount of treatment that patients need based on their individual risk. And not every patient is going to receive a stem cell transplant for the first time. And so this is my group and my mentor who's standing in the middle. And I just, again, want to thank the PCRF for funding this work, which we just submitted an additional NIH grant from the National Institute of Health on Monday. And that is to conduct the clinical trial that I just described to you. And there is absolutely no way that that would have been done without the funds from the PCRF. So I sincerely thank everybody on this call for all of their support. Thank you.
Oh, you're very welcome. Thank you for sharing that. This is really exciting stuff. And now I would like to introduce Dr. Ashley Plant. She is going to share her work on DIPG at Lurie Children's Hospital.
Can you guys see my screen okay? Well, thank you for having me. I actually see a lot of people I know on this call. So far I've seen John and Tracy and Michelle and probably Hildy as well. And I miss seeing you guys in person. I'm glad to be connecting with you virtually. And a lot of you have actually supported this project directly or other projects that I'm working on. So thank you so much for all of your guys' support. So I'm going to talk about one of my projects, which is immunotherapy for DIPG, and just kind of give some high-level information on that. So I'm 50% clinician, so I wanted to talk a little bit about COVID in the clinical world as well. So in general, the main problems that I see in pediatric oncology and in my world in neuro-oncology specifically is that pediatric brain tumors remain the second most common type of childhood cancer and also remain the #1 cause of cancer-related mortality. And there are still multiple types of brain tumors that don't have any curative options, DIPG being one of them. Our newest challenge is taking care of kids with brain tumors in the setting of COVID. So you can imagine those of you who've been through this or know someone who has, having to get some of those early discussions about diagnosis and even prognosis of your child's disease and only having one parent there at a time. Or if your child is sick and needing to be hospitalized, not having both parents being able to be present and having the siblings being separated is really a challenge. Also, all the COVID tests that children have to undergo on top of all their procedures and treatments that they're receiving just to be able to keep everybody in the hospital safe. So every time they get anesthesia, these kids are getting COVID tests. Every admission is another COVID test. Also, the inability to access the larger support groups because everyone's having to stay apart and socially distant. So I don't want to take away from the clinical challenges that we're seeing as well. My heart centers around DIPG, which is the acronym stands for diffuse intrinsic pontine glioma. In general, it's an infiltrative expansive tumor of the pons. So this is the middle section of the brainstem where all of our essential functions are. About 200 to 300 children are diagnosed in the US per year, and about the same amount abroad as well. One of the very difficult parts of these brainstem tumors is a lot of these children are fine until a few weeks prior to presentation, and then all of a sudden their child starts having issues with their facial movements not being symmetric, losing their balance, slurring their speech. They show up in the ER and get a MRI or CT only to find out that their child has an incurable brain tumor. So it is an emotional roller coaster even to start. The management unfortunately includes only palliative radiation, so basically radiation for the purpose of extending their symptom-free period, but we know that that is not curative in itself. And then families are forced to either decide to take their child home or to pursue clinical trials around the country, oftentimes leaving the rest of their family behind to try to find a treatment for these very difficult tumors. So right now, the median overall survival is 12 months, so just a year from diagnosis, and under 1% of children are alive at 5 years after their diagnosis. Fortunately, there have been new insights into the biology of this tumor. So for decades, we've known that this type of tumor exists, but haven't understood the biology behind it very well. And part of that is because doing surgery or even a biopsy on these tumors is very scary, to say the least. So some very brave surgeons took this on around 2010 to ask families to allow us to try to biopsy their child's tumor for the purpose of science to find new treatments for this disease. Fortunately, those biopsies went very well, and we also discovered that more than 80% of these tumors actually have a mutation in the same protein, a histone 3 protein, or histone 3 gene. The reason that's important is because these histones, as you remember from biology, actually help the DNA wrap, and it also can act as a conductor for which genes are expressed and not expressed based on markers that are present on the histone, either acetylation or methylation markers, which we've heard in other talks today. So this gave us a lot of insight, although this remains a difficult target because it's not something that you can simply find a drug to turn off its function and everything will go back to normal. So we have to be a little bit creative in coming up with possible therapies for this disease. My second passion after DIPG is immunotherapy in general. So the idea is using the immune system to kill tumor cells. And the idea of this is interesting to me because potentially that would offer long-term surveillance by the immune system for cancer cells. So a really long-term potential cure for them. The other area of neuro-oncology that's really hard is survivorship, because actually kids that we do cure end up with so many long-term toxicities of the treatment itself that their life is affected well into adulthood by their cancer. So that can include radiation effects, so neurocognitive functioning, the way they look physically, their ability to graduate high school and college. So immunotherapy potentially offers something that wouldn't have these effects in the long term on their quality of life. So I won't go into too many details on the current treatment strategy, but we actually are about to submit to the FDA our phase 1 clinical trial for immunotherapy for DIPG and another related tumor called diffuse midline glioma, which is a kind of childhood glioblastoma. So our current strategy is combining immunotherapies. So like normal chemotherapies or other targeted therapies, cancers learn to have resistance to our therapy. So we want to start out of the gates with a combination therapy that would look at both a vaccine therapy and then also that blocks the off signals to the immune system to allow it to ramp up even further. So for the vaccine, like you give vaccines to infections, basically we'd be injecting into the patient markers of the tumor. So we would call those antigens. Here it's a peptide that reflects a mutated protein within the tumor that's different than normal cells, and try to tell the immune system to recognize this as foreign and to go ahead and attack it. The checkpoint blockade, this combination therapy with anti-PD-1 and anti-CTLA-4 molecules, will also prevent the T cells from either being turned off because of exhaustion or being turned off by the tumor itself, which we know are forms of resistance within brain tumors. The other thing we're doing is trying to strategically place this immunotherapy right after radiation. So all the kids that are getting radiation will have these signals or markers of the tumor be exposed during the time of radiation. And then the vaccine and combination immunotherapy will take advantage of that situation to try to really ramp up the immune system to fight the tumor itself. So we think that this will be both specific because it looks at the markers of the DIPG and diffuse midline gliomas specifically and tells the immune system to attack them, and then also tries to overcome some of the potential resistance mechanisms up front to hopefully have the most impactful treatment for these patients with very poor prognosis from diagnosis. So impact of COVID on research in general, I know we've heard from Brian and Elliot on this. So funding sources is obviously a challenge. Our families have continued to donate even during this very difficult time, which we really appreciate, but some of our kind of standard funding approaches are a little bit unknown at this point. Also within the clinical world, all the non-interventional research studies were on hold for a period of time, and it's not clear what will happen with that going forward. So basically any trials that offer treatment for patients can still go forward, but biology or more investigational studies that don't have treatment options might be on hold for a period of time. Also, I collaborate with a lot of immunotherapy labs, and they take a lot of samples from our clinical trials to learn more about how the immune system is responding, and they have reduced capacity because, like we've heard before, only a few people can be in the lab at a time. Also, it slowed the review and regulatory board processes, so things like COVID-based trials will obviously have priority during this time. The FDA has done a good job of keeping up with the interventional studies, even within pediatric oncology, but that changed, or that structure has changed as well. And we just need to prioritize more and more which clinical trials we're pushing forward. So in general, we know pediatric brain tumor is a leading cause of cancer-related mortality in children. Generally need our help, need a lot of research still. DIPG is basically an incurable brain tumor for most patients diagnosed with this disease, and we hope that this tumor-specific combinatorial immunotherapy will be hopefully an efficacious treatment for them, and we will hopefully be enrolling patients early 2021. And though COVID creates a lot of obstacles for both research, our trials, and our patients, we definitely know, like everything, we won't let it stop us, and we'll continue our work here. And I definitely want to say thank you to all of our supporters, Will Strong Foundation, the Irwin family, and PCRF for their support. We also have other families that I think are on the call too. The Liam Bear Strong Guild Family Fundraiser raised money for this trial as well, and we're just super grateful for all of the support. And during a time like this, it really means a lot. So thank you.
Thank you, Dr. Plant. Always nice to hear you talk about that, and I wish you best of luck. I know getting this FDA across the finish line has been a huge Herculean task, so thank you for your continued effort. And with that, I would like to introduce Katie, and Katie's going to talk a little bit about working with our families with the brain tumors in the children and what her job looks like right now.
Thanks, Jerry. Thanks everybody for having me and allowing me to join today. Happy to be here and to be joined and invited into this group of our researchers. In lieu of a slideshow, you'll have my face here today. But I wanted to share a little bit as a social worker. I am the brain tumor social worker at Lurie Children's Hospital in Chicago, and been asked to speak a little bit about how patient care has changed and how the experience for our patients and families has changed throughout this pandemic this year. You know, when I've had folks ask outside of my work, you know, how our patient families are doing and how they've been coping through this pandemic, I've shared that, interestingly, in the early days of you know, COVID-19 and maybe February, March, April, when talking with our patients and families about how they're doing, a lot of them had the same reaction, which was, you know, as we were in the beginning stages of adapting to this, was, you know, we're doing okay. This world of wearing masks and washing our hands and using precautions and limiting family members coming over during flu season, this is our world. This is what we've always done. So they've been used to coping that way. They, I think, if anything, were a little bit affirmed by having the understanding of, you know, their community, getting to see a glimpse of the things that they were already doing day to day to keep their families safe and keep their children safe. Obviously, like most of us, over time as we endured more and realized that there wasn't sort of an end in sight to this pandemic, we all grew weary of some of those changes, as our families did as well. What started to become challenging more on the hospital side for our children who are in treatment, for our families, is some of the policies that we were working on putting in place, universal precautions to keep our patients and families safe, to keep our staff safe. So Dr. Plant referenced things like only being able to have one parent or caregiver stay bedside with their child, either during clinic visits or during treatment visits. Navigating that through, new diagnoses, patients coming in newly diagnosed for the first time and trying to explain the intensity of the treatment that we would suggest for their child, or maybe on the other end, preparing a patient and family for end-of-life care and what that might look like and how we could adapt within our hospital environment while adhering to our new policies, but also honoring and looking into exceptions that could be made to support patients for having, if there's two parents in the household or a sibling, come to be around and spend time with them. I think about, during the pandemic, the first experience I had with losing a patient and thinking about how that changed the way as a social worker that I was able to be there to support family. A lot of times, as many people are familiar with, if we have a family or a child that may choose for their end-of-life journey to be in our hospital, we're able to surround them with their family, their siblings, their parents or caregivers, to support visits from a favorite teacher, from a friend, from a pastor, and looking at the way that we would have to now modify that for families, and trying to look at how we could still provide them with the peace and the comfort that they would want for their child, for the things that patient would want, look much different. I think about that first patient I remember passing in late April, and thinking about even just the nature of interacting and talking to a person, doing things that I now consider differently in the age of this pandemic, such as even placing a hand on their shoulder or giving a hug to comfort them, even spending time sitting and visiting and talking with them face to face. All of this is, now changed throughout what we've started to look at as new precautions for, infection control and social distancing. It's certainly changed the nature of my work in terms of, in the early days thinking, okay, do I need to limit some of these visits face to face with families? Can I call and have the same conversation with them over the phone about how they're coping, what their family's needs are, how the other children are doing in the home? And while we certainly were able to do that, and we've all adapted to things such as this, virtual calls, it's different. We're all in a helping profession, are used to working with people face to face, and it's much different to do those things virtually. I'll fast forward a little bit towards, through summertime and now into fall, what we're seeing as a big challenge, in addition to our families who are already coping with childhood cancer and they're coping with COVID-19, is getting back into school. So school's a hot topic for a lot of our families right now, trying to figure out the world of e-learning or hybrid learning, navigating school while still continuing with treatment for their children. Walking down the halls of our clinic, it's common now, looking in every room, there's a tablet, there's a Chromebook, there's somebody's teacher on the other end of the computer while they're waiting to be seen by their doctor in clinic. Patients trying to navigate having the school's support and accommodation and having cameras off because they still want to stay engaged in school even while they're receiving chemo or they're in the hospital. Having schools understand that our patients still need accommodations just as usual, even though they're home doing school or able to open their computer at home or in the hospital, but providing education to our schools that our students and our patients still really need the support, that there's still those days just like normal that they really aren't able to do school. They're not able to physically or emotionally or mentally, even though they can open their computer instead of go into the classroom, but that they still need those same accommodations and even more. A lot of our patients, many of our teens especially, are just expressing how isolated they're feeling, which many of us I know can relate to, but on top of this pandemic this year and going through treatment, missing out on some of those things that would otherwise feel normal to them. So like I mentioned, going to school, or even some of the things that, you know, our patients look forward to, planning for their Make-A-Wish or for trips or special occasions, birthday parties, even events in our hospital where we have special visitors come, we have pizza parties, or our patients are able to gather in the playroom or in the teen lounge and connect to each other and speak each other's language and support each other. Obviously, many hospitals are now putting precautions in place for safety, but with that also comes then that isolation and that disconnect. And then Dr. Planino also mentions many of our families that may travel state to state, may travel across the country to access some of these clinical trials that we have discussed. Looking into, okay, how can I safely travel with my family? How do we travel? Do we take a plane? Do we drive? What feels safest for us? Where do we stay when we get there? Looking at different, travel orders state to state and different policies between hospitals and figuring out there's nothing that our families wouldn't do for their children. Yet we know this year there's even more barriers in place than we might normally expect. So there's been a lot of support for our families of trying to ensure they can safely travel and have safe accommodations, and also support then, you know, if that's just one parent coming instead of the whole family, that separation in that family unit, what that looks like and how that may add additional strain to our families. And another big discussion that's been happening is families' concerns about insurance. So obviously with this pandemic, lots of our families are experiencing loss of income due to either losing a job or not having the ability to work from home and help their children with school if they're e-learning. Also thinking about then changes to insurance that might be upcoming, looking at concerns with if there's changes to pre-existing condition coverage and insurance, and prepping for what that might mean financially for their family, or fearing if there would be ever loss of treatment for their child. So like many on this call are familiar with, our patients and families go through the unimaginable. They go through lots of stressors and draw on an amazing amount of resilience to cope and to support their child through treatment. This year, we know that the amount of those stressors has been layered on top. My biggest takeaway and what I'll end with is that we know that our patients and families rely on us. They trust on our care and on our expertise. I think now more than ever, all of us truly are looking for a sense of grounding and stability to help us get through this time. But our patients and families, more than ever, are really looking to us to stay the course, to keep providing the care that we provide, keep funding and working on research so that we can continue treating and curing childhood cancer. Thanks for having me.
Awesome. Thank you so much. I'm just so honored that all of you guys took this time. We have the rest of the panel here. I think, Katie, even some of the little simple things which are huge is that I'm sure you're not having patients and families ring that bell when they finish their last treatment, and the pet therapy, the music, like all of the things. All of us know that are in this world that there's so many more components to take care of a child in a hospital versus adult care, and I think that's you gave a very touching view of what it's like. Our researchers are fantastic, but you are also there for all of these families, and your support is invaluable to the rest of the researchers and families, and I totally appreciate you taking the time to do that.
Thank you.
We didn't have any questions right now. I know we have quite a few people on here, but I think that obviously this is a lot of information, a lot to process. I did just want to echo on really what all of the researchers have said, and starting with Brian, is that we as a funder of research are all very concerned of what the future of research is going to look like, especially for the emerging folks coming out of the block. They put in multiple years of education and schooling, and then all of a sudden here we are, and the funding is just not there. All three of our researchers we have on this panel are well funded, but I would imagine, and any one of you can touch on this, that there's still some apprehension of what the next couple of years is going to look like. And, you know, anything any of our organizations can do to help, you know, we're going to be there for you. But I'll open it up to you if you have, you know, what are your biggest fears right now, or what are your biggest joys in what the work that you're doing.
I mean, I think one of the things I'm most worried about is it's not my idea or my original thought, but I've heard a lot of more senior investigators than me talk about a concern for like a lost generation of trainees, where there's already a lot of pressure for, you know, these like MD and PhD or MD PhD postdocs, these trainees who don't have their own research labs yet, and are trying to figure out what their thing is going to be. And, you know, there's already a lot of pressure for them to go to work in industry, which, I mean, the industry does a lot of great stuff. Like they bring all our new drugs to market, and that's totally invaluable. But especially in pediatrics, where all the great ideas have to originate in academia because of the rarity of the diseases we work on, losing, you know, 5 years worth of trainees will be devastating, right, to the science and to the progress in the field. And you know, so a lot of us are worried about that, and I definitely am one of them. I mean, the postdocs who are in my lab are in a semi-fortunate, a relatively better position because they're new enough that they're not going to be on the job market next year. They're still working in their projects, and we got lucky with some funding that came through just before, you know, we actually had to go home. So they're sort of in a nice cocoon where I can keep them safe for now. But there are many who are sort of right at the point where they should be jumping off and starting their own labs. And there's not, I mean, almost every institution has a hiring freeze right now. And so, you know, I think, I don't know what the fix is, but I think we need to be, the other thing that I think others have pointed out is that, you know, our meetings now, which used to be these like sort of fantastic, huge extravaganzas, especially our biggest meetings where, yeah, sure, the famous Nobel laureates gave a plenary or a keynote, but there were a lot of little small meetings where postdocs could go and show their groundbreaking emerging work. You know, basically what those meetings now become are Zoom meetings with only the plenary and the keynote, and all the other little stuff is gone. So now there's no opportunity for the trainees to network, and no opportunity for them to showcase themselves as the future leaders of the field. And so we're going to have to find some ways to fix that. But I think, you know, just because this is a foundation talk and because of all the amazing work the foundation does for sort of helping emerging people transition to independence, I mean, I think that from a funding perspective, there's a significant vulnerability specifically for that group.
Well stated. Well stated.
I hope that there are some silver linings in this relatively dark time. I think one thing you heard from all of us is that when the shutdown happened in the lab and in the hospitals, we all tried to double down on analyzing data that we already had in hand and sort of finish up as many projects as we could. And then, you know, I think the big question is how long is this pandemic going to go on going forward, which is going to dictate what happens with research. But there are a few bright spots. I think one is that just like we're doing this Zoom meeting instead of in person, our patients are now getting access to remote healthcare in a way that they didn't before. And so, you know, we used to ask patients to come from 3 hours away for a routine follow-up that now in retrospect seems, you know, so archaic and barbaric in a way. We can do the lab close to home and we can talk with them. And if there's anything that needs to be evaluated on physical exam, that's a separate situation. So that's here to stay. We're doing a lot more remote second opinions, for example, now that insurance has caught up and mandates reimbursement for those. So I think, you know, there will be access to care that's a little bit easier for some of the more remote patients. And like Katie said so eloquently, I mean, a lot of our patients have been dealing with these restrictions for a long time. And in a sense, it almost evens the playing field. You know, our kids are actually going to school more frequently now than they were before the pandemic, because these virtual opportunities are present that were not there before. So I'm holding out a small amount of hope and looking at the silver lining as well.
I like that. Ashley, did you have anything you wanted to add before I wrap everything up?
Yeah, just quickly, I mean, I worry about our families, and I know there are a lot of families of patients on the call too. It's just the normal support that we could provide for them during difficult times is a little bit limited by COVID. And of course, we're always amazed by how our patients and families pull through anything and pull through anything together. But just remembering, if you know somebody who's still going through treatment or you're a part of a support group, to really reach out more than ever, even if it's just virtually, to provide that support. I do think, you know, we haven't been a priority of government funding before, yet this is a great example of how we pull it off anyway. So I think we're still going to be fighting the fight, and we'll all pull together to do this. But just being aware of those who might be struggling more than others to, you know, keep that community going.
Awesome. Well, I can't thank you guys enough. It's always nice to get caught up with you, and this does afford me the luxury of staying in touch with you guys and then sharing this with our families, our board members, and our constituents. And I'm just really incredibly grateful for the work and for you guys, you know, taking the time to be with us. It's been an amazing afternoon, and I wish you guys all the best and continued work. And thank you everyone for joining us, and know that PCRF is not going anywhere. We are going to continue to fight. I just want to leave these last two images with you. The young woman on the left, Sylvia Escalero, is in medical school now at University of Michigan, and she's a childhood cancer survivor. She's one of our scholarship awardees, so I love showing her picture. And then we have Mia the Brave, who is Will Irwin's ambassador this year, and Mia is on her second round of cancer, and she is fighting it, and like I've not seen another child do. And so that is why we call her Mia the Brave, and she's also an ambassador for PCRF. So I thank all of you for your support and your time, and we'll have this recording, and please feel free to follow up with any questions. And everyone have a great day, and thank you so much.
Thanks to all of you. Thank you.
Bye.